Hello and welcome to to this webinar. We've been looking forward to this. Diana and I, we're going to be celebrating PRO-C3 that of course was put on the Roche Alexis equipment and is now available in Europe, later also in the US, done with our fantastic partner Roche. But we're going to talk about fibroblast activity and PRO-C3. And we've been working hard on putting all the best data together, and I hope you will enjoy that. You can always ask questions to the speakers through the private chat, and those will be answered in the Q&A session. So for those that do not know us, my name is Morten Karstel. I've been at Nordic for 27 years, and I'm the head of R&D. I'm super proud that I have 700 publications on PubMed and an H factor of 103. And if there's one thing that I have learned from all those publications is the fact that we need collaborations. We need good collaborations to move anything, such as the Roche collaboration, such as all the collaborations that Diana is going to show you on the timeline of PRO-C3, because this has been an army of research and so many different people joining forces in moving PRO-C3 forward. So we're going to be celebrating a lot of people today. And talking about Diana, Diana is also a very accomplished scientist. I think I've been working with Diana for 21 years. She has an H factor of 67 and more than 200 publications. And Diana is of course going to tell us some of the best highlights on PRO-C3 in liver and in lung. So I'm going to start giving a little bit of introduction to Matrix and how we see the world. Then Diana will give us a timeline of PRO-C3. I will show you a little bit of cancer, some of the most exciting stuff we have seen, how dangerous fibroblasts are. Then Diana is going to do lung and liver. I will add a few other diseases in which fibroblasts are important, and then we'll wrap up for questions. And we've really been looking forward to this, giving you the highlights of our research from the last 15 years. Actually. Is it important? Is it an important disease we're looking at? Yes, it is. It's actually around 40% of patients in the Western world that are dying from fibrosis. And I think it's a little strange if you go to the doctor until now, you're going to measure your cholesterol, your CRP, but we have not really been catering to fibrosis until now. And I think we need to be catering to that, so patients know what is their level of fibrosis activity. Actually, 1 in 4 live with fibrosis, and you can see that there's increasing prevalence of fibrosis over the last many years. There's actually more than 150 different diseases with fibrosis, and of course some are bigger than others. So we've been putting all these data together from a Danish data source, and we are validating in the UK Biobank. And I think it's really, fibrosis is big. And we need to cater to those with fibrosis a little bit better. So of course, fibrosis is in the center of our heart, and now with Madrigal and Novo having approved therapies for MAFLD or MAFLD, then of course we'll be celebrating that at the Extracellular Matrix Pharmacology Conference next year. And here, matrix pharmacology is at the center of all things. We'll be discussing matrix and lung and liver and skin, kidney, cancer, intestines, and of course autoimmune diseases, really extracellular matrix pharmacology. Last time we were 450 people, it was 180 abstracts, more than 80 presentations, and really a lot of fun. And I'm promising good weather. This is not a Nordic conference, I'm just the proud chair. It's actually a non-profit society that's hosting this, so we need good speakers and good abstracts there. Well, so extracellular matrix, is it important? Of course, I think if you are in this webinar, then you also think extracellular matrix is important. There's actually more than 10,000 publications now on PubMed every year on matrix. If you look at how fast it's growing. There's 185% every year more matrix publications. And so if we look at different organs, and particularly in fibrotic disorders, there's actually up to 75% of that tissue that's actually matrix. So of course, matrix is important. We know that it determines the extracellular matrix, or the extracellular matrix determines the fate of cells, the shape of cells, the function of cells, and therefore, of course, we need to understand what's ongoing around the cells. So taking a little bit deeper dive into the matrix. And today, of course, we are going to be discussing mainly type 3 collagen. We are going to see how that dangerous collagen, that's a fibril collagen produced by the fibroblast, is blocking organ function, leading to organ death and leading to the death of patients. I think it's very important that we measure biomarker that's related to outcome of patients, because if it's not related to outcome, then how can we know it's important? So of course, we're going to be talking a lot about fibrillar collagens, dense fibrillar collagens that are impermeable. Those collagens are impermeable to oxygen in the lungs, to blood in the liver, and it's really changing the organ for the worse. Of course, there are many other collagens. There are 28 with 40 different side chains. But today we're not going to be discussing the beautiful collagens that lead the collagens of the basement membrane, those that are permeable, those collagens that are loose and allows diffusion of oxygen and species. And so on the top here, you have epithelial cells. They are smart cells. They know which way is up and down, and they have a specialized matrix. And on the bottom here, you have fibroblasts. And I think fibroblasts are a little stupid and aggressive, and I'm afraid of fibroblasts. And I think after this, these slides Diana and I are showing, I think you will be afraid of fibroblasts as well. We're going to show a little bit of type 6 collagen. There's a new collagen hormone in town called endotrophin. It's going to be very important, and it's even more related to outcome as compared to fibroblasts. And we just show a little bit of that today because there will be it's an important molecule, and it's even more related to outcome as compared to PRO-C3 and type 3 collagen. If you need a good Christmas present, then I recommend the collagen book. It's all science, really talking about matrix. And then, of course, we have already talked about the extracellular matrix pharmacology conference. So what we are doing at Nordic is a little bit different. And I'll come back to the hypothesis-driven approach that we have taken to develop biomarkers such as PRO-C3. Because we think that and that was the lesson we learned in bone that I will show you in a second, that in a healthy tissue, there is a balance between tissue formation and tissue degradation. And if there is more formation than degradation, then we actually get fibrosis. If there is more degradation than formation, we have inflammation, we have autoimmune diseases, we have bone loss, and we have other diseases. So it's very important when we quantify something that we actually know what it is we are quantifying in serum. We need to know if that fragment or protein we are quantifying, is that related to formation of the tissue or degradation of the tissue? And we need to know if our pharmacodynamic measurements is good or bad for the tissue, and we'll show a lot of data on that. And in fact, we think that you can divide most diseases into a balance between extracellular matrix degradation and extracellular matrix formation. And of course, I've just mentioned bone. We are going to be talking a lot about fibrosis and other diseases, but my favorite example is actually Crohn's disease. 5 years ago, everybody thought that it was one disease, but now we realize we have a fibrostenotic disease where we have much more formation than degradation. And there is a fistulizing disease with penetrating disease that's devastating where it's inflammation. So sometimes we can see that the same disease is actually two different diseases, but in one of the diseases that's much more formation, and in the other one it's much more degradation. And of course, we need to target the treatments to those molecular endotypes. So we've been looking at biomarkers for a long time, and today we are focusing on PRO-C3, and we're talking about fibroblast activities. And so why should we actually be working with those biomarkers? Well, We can use them to identify patients that's more likely to respond to treatment. We're going to show you some of these things. Fibroblast activity is a central component of response to therapy in a range of diseases. I think you will be surprised today. Of course, we can assess the drug's efficacy in a shorter time frame versus standard technologies in the liver field. Of course, some compounds change PRO-C3 levels already after a week, where you need a year or possibly more for seeing an effect on a liver biopsy. And of course, the only way of really finding the right and optimal dose is through the use of pharmacodynamic biomarkers and really understand the dose optimization in those early proof of concept studies. And then, of course, giving us that insight into the critical path of the disease and the drug mechanisms of action allows us better to understand and believe in the compounds. So we call that a drug development filter that we proudly use together with many of our collaborators. So clearly, there'll be clear go-no-go decisions. We have a higher chance of success in clinical studies. And then we see an efficacy shift. We can see efficacy earlier, possibly already in 1B. We'll see a little bit of those data. And then, of course, that allows us to direct resources to our place. So basically, I think what we can do with these biomarkers, if it's a good biomarker, and I'll come back to what I think a good biomarker is, and we are writing a few publications on that. That's a biomarker that allows you to look into the future. And that means you can get the data in a phase 1B study, for example, that you would normally only get in a phase 2A or 2B. Looking at a pharmacodynamic, a surrogate measure of efficacy for liver fibrosis, kidney fibrosis, skin fibrosis, or autoimmunity allows us to believe in that compound. And I think we need to measure those biomarkers because all these different organs, they're composed of matrix. And to be provocative, I don't think we are truly efficacious if we are not changing the matrix. Because then we're just symptomatic. We need to change some of that matrix back. And I think in the liver field that has been wonderfully done for some of these patients. So we can change the matrix, and we should change the matrix, and therefore we need to quantify that. So of course we've been lucky with a range of biomarkers where we can do this, and that allows us to look into the future. And today we're going to talk about PRO-C3. So what is my favorite biomarker in the world? And many have helped me coin this terminology. And we have a paper coming out, and it's a unicorn biomarker. So what is it that a unicorn biomarker is able to do? Well, to me, it's a biomarker that is diagnostic, and it means elevated in those that have the disease. It's at the same time prognostic, meaning that those that actually are progressing, dying, they have higher levels. And then it's pharmacodynamic, meaning if we have a compound that's actually working, that will go down. To me, that's central. And that leaves very, very few biomarkers on the table. Then if we're lucky, they're genetically validated. They need to be regulatory supported. I'll come back to that. We need to have them scientifically understood, as FDA is telling us. In order to be a reasonably likely surrogate endpoint, you need really to understand your biomarker. It needs to be in the critical path central for disease. And of course, they need to be technically validated, and I'll come back to that. And so that means there's not a lot of biomarkers left. So what is my favorite unicorn biomarker, my favorite biomarker in the whole world is HbA1c. I think it's interesting because if we have measured, or if we are thinking what is hemoglobin a biomarker for, and we had done a standard array, we would actually have seen that would be lower, and it would have been a diagnostic biomarker with a lower level for anemia. Now it's a special glycosylation, and that means it's the best biomarker for diabetes. And that means it's diagnostic, it's prognostic, it's pharmacodynamic, it's even a surrogate endpoint. You can have a drug approved on this biomarker. So there are some unicorn biomarkers out there that is really working and helping drug development. That also means that we think it's important not just to quantify the total protein, because of course, total proteins have many different domains. We accept that there's a catalytic domain, there can be a pro-domain, there can be other domains. Of each of those domains means something different. So we think, and I think we have proven that for many of our publications, that quantifying a specific fragment with a monoclonal antibody in a given assay is providing superior value. The cognitive field were there many years ago. They knew that that amyloid precursor protein 1,2,44 was very different from 1,2,42. So we know different fragments can be either good or bad. They can be pathological. They mean different things. And at least they enable us to separate tissue formation from tissue degradation. So actually, we've measured many, or we have developed many assays, but we have not one assay that's measuring a total protein, only fragments of proteins, those that have pathological relevance, just as HbA1c. I wanted to just give a little credit to where we come from. So these are some of the wonderful data done, I would say, a long time ago, more than 25 years ago. And so on the top, this is a phase 2 study with alendronate, the first drug that got approved for osteoporosis. And what you see is a sharp decrease in bone resorption. Actually, 75% decrease in bone resorption. That, and then on the lower part here, where you can see bone mineral density of the lumbar spine, and that's of course a much slower difference. It actually takes more than a year before you can see a significant decrease on bone mineral density. That means that the bone field changed. When this was developed, the bone field changed because then there was a development of anti-resorptive, those that blocked bone resorption, CTX1. Then later came the pro-anabolics, those that stimulated bone formation. And I think we need to adopt the same terminology in the fibrosis field. You cannot just be anti-fibrotic. We should either be anti-fibrogenesis or pro-fibrolytic. If you're both, congratulations, it's a fantastic compound, but we need to assess those two processes individually in order to communicate what is actually the mode of action. Clearly, these biomarkers changed the bone field. I was happy to be part of that. We put them on the Roche equipment a long time ago, but they continue to be pillars in our understanding of biology. So each protein fragment actually tells us a different story. This is type 1 collagen. Type 1 collagen formation can be measured by the propeptide. Then we have bone resorption. That's a different fragment in type 1 collagen. It's actually a cathepsin K degraded fragment of type 1 collagen. Since CAT-K only is an osteoclast, it's bone specific. Because it is interesting, and I'll come back to that type 1 collagen is relatively unspecific. And then there are other fragments that provide different things. So it's important with this neoepitope assessment. It's important what we are actually measuring. Is it the total protein? Is it the proform? Is it degradation? Is it some kind of cryptic signaling? We've developed specific assays for collagens that are signaling, in particular tumstatin, vastatin. Is it polymerization? For example, CRP is actually a pentamer. Or is it one of those single amino acid post-translational modifications, just as HbA1c, but it can also be citrulline dimension, it could be others. I think really our eyes are being opened, and in order to move modern clinical chemistry forward, we need really to assess the neoepitope or the epitope. So I think it's wonderful thinking of biomarker development, and then also thinking of the biomarker valley of death. So there are many similarities between academia and industry. We all start with a great idea. I'm going to come back to how we do this hypothesis driven. Then we can create a candidate biomarker. We make an assay. Then we do a technical validation. Then we measure some samples. Hopefully we get a fantastic publication on that. But then often the academic world is stopping. But then the real work starts. This is the biomarker valley of death. You transfer that to a production. That production takes a reasonably long time to make a fantastic assay, and then you need to do all the studies again. You need to show the biological relevance, and then you have a fantastic assay you can use with your collaborators. But then commercialization, that's where we have fantastic collaboration with Roche Diagnostics. I mean, we need to have regulatory approval, we need to have a context of use, and it needs to be commercialized worldwide. So in this pathway for biomarker development, and it will be described carefully in our unicorn biomarker paper. Clearly, there's a valley of death from where you have a fantastic discovery, then it takes 5 years before you actually have a true assay that can be used on a platform that goes wild world. And that's why it's important that we invest in biomarkers and that we push them forward together. So really, it is a strong, or it is many types of research that needs to be done to push a biomarker forward. We need scientific, technical, and regulatory excellence. Nordic Bioscience started 30 years ago, and we had 3 biomarkers, FDA approved, CTX1, osteocalcin, P1 and P. We're proud of that. But just talking about PRO-C3 today, we have had 3 different letters of support, of which PRO-C3 is one. We have submitted PRO-C3 with a letter of intent to FDA. We have a qualification package advice. We have sent in a full qualification package. Daniel, if you're listening, thank you for all your work. And then we had an EMA critical path meeting. And so it takes a lot of time, and there are many different regulatory interactions that needs to be done. Data needs to be generated. Data needs to be crystal clear and perfect. And we'll come back to some of those things. And so I think it is important that FDA supports collagen biomarkers. We got a prognostic enrichment of solid tumor letter for PRO-C3, and I'll show some of these data, and then actually also prognostic enrichment for heart failure preserved ejection fraction with PRO-C6. So there is a regulatory path. And then of course, when we are measuring these biomarkers, it needs to be done in the best environment. And sometimes we forget that. Sometimes we forget that it is an art form to measure a biomarker in the correct environment. And so we are super proud that when we do proficiency testing, when you're supposed to hit a number in your lab, then we have the highest that I know of accuracy for this proficiency testing. And then we deliver, of course, good data to our clients and collaborators. And so we have this CAP-PIA certified lab that's part of the high quality that is needed for, among other things, PRO-C3. So how did we do this? I mean, we didn't do an array. We do things a little differently at Nordic. We really develop hypothesis-driven biomarkers. And so we are experts. I think my team is one of the best ones in the world for extracellular matrix biology and the quantification of extracellular matrix biology. So we have a hypothesis, we develop a prototype biomarker that gives us some biological understanding, and then we do a real assay. And of course, that hypothesis-driven is based on extracellular matrix, and that's of course essential in IPF, in SLD, chronic kidney disease, fibrostenosis, cancer, and heart failure, and other diseases. And it provides a mechanistic relevance to start with. This collagen is important, this protein is important. I think it's also easier than that to be on the critical path of disease. We're not just measuring many biomarkers, we have selected some that we think are super important for the disease. And then we validate them in translational model. And I'm going to come back to PRO-C3 and show really when we measure PRO-C3 in serum, it's really a serological measurement of fibroblast activity. So how do we link those cellular activities to serological activity? And just going back to type 1 collagen, because type 1 collagen is the most abundant protein in the body. It's by far the most abundant protein in the body, and so how can that be specific for many things? So that's also why it's not a good fibrosis biomarker. But these special fragments are making it specific either for bone formation or bone degradation. Same thing for type 3 collagen. We spent a lot of time trying to understand type 3 collagen. It's a fibrillar collagen, which proteases are cleaving off the propeptide, which different domains mean different things. And so we actually developed more than 30 different assays for type 3 collagen. Some of them you possibly know, CTX3, C3M, but we've developed many assays for type 3 collagen that each have a biological meaning. And of course, PRO-C3 is the exact place where ADAMTS2 is cleaving the propeptide off. That allows the fibril to be made of these dangerous collagens that later become fibers. So when these fibroblasts becomes activated, and only activated fibroblasts actually produce type 3 collagen. When you super activate a fibroblast, they will produce 100% more type 1 collagen, but they will produce 1,000% more type 3 collagen, and that's why it's much more accurate for fibrosis. Then we can measure these biomarkers of cellular activities in the blood. And so before we venture further, Diana is going to take you to the history of PRO-C3 and I think say thank you to 1 million people.
Thank you, Morten. And so I'll just walk through timeline that Morten mentioned. So it's linking a bit back to what Morten mentioned, that it actually takes a long time to produce a biomarker into a high quality, and it also takes a lot of efforts, and that's from many different teams around the world in this case, that we really pitched in to develop the PRO-C3 timeline. So I'll start out with the first slide here. So I have 2 slides on the timeline of PRO-C3. So we actually go all the way back to 2008, where we started to think about the design of this target that we have for PRO-C3 that Morten just presented. So we designed the target for the antibody here in Herlev in Denmark, and then we generated the antibody in Beijing in China. And that took some years until 2010 really to do all of this work. And then when we had an antibody that we approved of from the screening that we did, then in 2013, we actually ended up having the first PRO-C3 assay developed and the first publication on this. Again, many people are on the slides, and there's many more to thank for really the development of PRO-C3. So you'll see names here, but you know, we could add in many more names for each of these milestones in the PRO-C3 development. Then if we go to 2013 and 14, here we really start to publish the first data on PRO-C3, both in preclinical models, showing that actually PRO-C3 comes out of fibroblast and also can be used in animals and also in patients. So these were like very important data to start to publish, and we show them at different conferences, really stating that PRO-C3 is relevant in this case, especially in chronic liver disease, but also in multiple fibrotic indications. Then in 2015, we started out the first kit productions of PRO-C3. So the first kits, of course, were produced as well, but when we went into the kit production, it's a kit production facility that we started to produce PRO-C3 when we had established the assay itself. Then we had a large milestone in 2019 where we actually published on the score called the ADAPT score, or the ADAPT algorithm, which is really the score that is now on the Elecsys cobas platform. And this was a milestone publication done by efforts from many different academic institutions and also together with Nordic Bioscience. So this was really something that moved forward the use of PRO-C3. So if we go to the next slide. Then in 2018, we actually also measured PRO-C3 in multiple IPF and cohorts and COPD cohorts, and showed that actually PRO-C3 was especially relevant for IPF, which is of course a very fibrotic indication that it manifests extensive fibrosis onto the lungs. I'll come a bit back to that later in the talk. So we did quite a few publications with great academic institutes and also with GSK in this case. And 2019, we did the first GMP production and CLSI validation, so a high level of technical validation of the PRO-C3 ELISA assay. And 2019, so you see we're moving a bit faster now. Here we actually then from 19 to 2023, then we transferred the PRO-C3 to the cobas Elecsys system. And again, taking a lot of efforts from multiple stakeholders at Nordic and at Roche and also at BMS. Then in 2020, Morten, he already touched upon this, we got a letter of intent accepted from the FDA for MASLD. This was done together with the Litmus Consortium, and also Nimble had a letter of intent including PRO-C3. So this, of course, is a very big milestone, showing that the FDA, they acknowledge this biomarker as an interesting biomarker to move forward. Also in 2023, we had a letter of intent accepted for the use of PRO-C3 for tumor fibrosis, again done by a big team here at Nordic Bioscience with efforts from academics providing some of these samples needed. And at this time point, we are above 100 publications, at least for PRO-C3, in multiple indications, especially in chronic liver disease, but really in multiple fibrotic indications. Then this year, in May, we had the PRO-C3 launched on the Elecsys cobas platform. Morten also mentioned that in collaboration with Roche, and also Litmus actually provided the samples to do this. So yeah, back to Morten, and I'll come back a bit more later on the liver and lung aspects of PRO-C3.
I think that was very nice, Diana, and a lot of people have helped us on this path. So I'm going to continue a little bit on with some of the most surprising data that we have, and I think some of you may know PRO-C3 and fibroblast activities, but this is really an area that has surprised us. So of course, solid tumors, they actually represents very big foci of fibrotic activity. There are many so-called cancer-associated fibroblasts. Pancreatic cancer may be the most fibrotic disease at all, and there are many reasons that's bad. It's limiting, of course, penetration of drugs into the tumor. It's also fueling cancer cells with different factors, making them more aggressive. And so many of these solid tumor types, they have tons of fibrosis, and that's important for how patients are responding. There are quite a few very good publications demonstrating this, and I just want to highlight all the different groups that have been pioneering, saying cancer cells are important, but possibly the cancer-associated fibroblasts around them are equally important. So one of the first letters of support that Nordic had was from FDA, saying it's a prognostic enrichment biomarker. We measured 2,000 different cancer samples in breast and pancreatic, in melanoma, colorectal, and liver, and we saw that independent of disease, there was really an odds ratio of 2 for progressing. It means the healthy reference range of PRO-C3 back then on the ELISA assay was 10. And then if your level was 20, then the chance of dying was twice. And since in cancers, the level is actually above 100, this is really a good biomarker for outcome. And so FDA is suggesting to use this biomarker for prognostic enrichment. And then we went on. So we measured different studies with different collaborators. And here you see 3 different studies. One is in non-small cell carcinoma, the lower one. Then it's metastatic colorectal cancer, and then it's metastatic breast cancer. And this is one serological measurement before treatment. And what we can see is, and we just divide this into the median. So the data you can screw the data to look even more impressive than this. If you have a high level in the breast cancer that are treated with trastuzumab, then your level or your chance of survival is actually 7-fold or 6-fold lower if you have a high level of PRO-C3. So PRO-C3 and fibroblast activities, super important for who's actually responding. If we look into colorectal, it's 2-fold. If we look into non-small cell lung carcinoma, it's 4.5-fold. So really, those patients with a high amount of fibroblast activity, they unfortunately will die. But the ones that have low levels, they actually have very good survival here. Let me show that in a different way with different studies. So this is all malignant melanoma. It's treatment with Ipi, Nivo, and Pembro, the 3 big IO treatments on the market that are saving patients' lives. And let's look at the middle one and the right one. This is it's smaller studies, but they are all showing the same thing. And this is again cut in median. And so if you have a low level of PRO-C3, congratulations, you have low levels of fibroblast activity. You are most likely going to be a very good responder to IO therapies. However, if you have a high level, then you have a worse outcome, and even worse than that, it's going to be fast. So if you are not afraid of fibroblast activities before this talk, I hope you're getting increasingly interested in measuring PRO-C3 or another fibroblast biomarker, knowing the fibrosis level activity of these patients. Another way of showing that these are the data, this is a different study with trastuzumab, and again it's a medium cut, and what we see here is that patients with a high level of PRO-C3 again, that they have a 5-fold lower survival as compared to those with a low level of PRO-C3. So high levels is bad. And then of course this is undisclosed with a collaborator. We asked them the question, what happens if PRO-C3 increase or decrease? Well, In fact, this is a combined chemo and antifibrotic. Those patients that increase in PRO-C3, they actually die faster, and those that decrease, they have longer survival. I think this is super important. I'm not saying that PRO-C3 is a signaling molecule. I don't think it does anything. It's just a signal that fibroblast activity has been there. But I think it's very powerful with the other data showing that when PRO-C3 goes up, there is more outcome, and when it goes down, there's less outcome. I think that's important data. And of course, this is one of the well-known compounds in the cancer field, the tyrosine kinase inhibitors. We've done this in a wonderful collaboration with BMS. And as you can see here, then PRO-C3 actually goes down in response to a tyrosine kinase inhibitor. It does not really respond to anti-PD-1, but we would not expect. It's a different mode of action. But for tyrosine kinase inhibitor, we actually see that there's a 20% decrease, and that, of course, associated with longer survival of cancer patients. But just 2 slides, that type 3 collagen is not the most important thing in cancers. It's actually type 11 collagen. So if you look at which collagens are these cancer-associated fibroblasts actually expressing, and there's wonderful work from Nicholas's group and Jeppe. And so you can see that healthy fibroblasts, the blue ones, they are producing a range of collagens. But the cancer-associated fibroblasts, they are exclusively expressing type 11 collagen. Type 11 collagen is not produced actually by other cells. And if we measure in serum, this is RNA-seq from many PDAC patients. If we measure in serum, then type 11 collagen actually has an area under the curve of 1 for diagnosing all solid tumor types. That has a lot of implications for antibody drug conjugates and other things, but just saying this is a PRO-C3 celebration. That's just remembering that there are other collagens. And so I want to take you back to cellular activities and translational science. We do a lot of translational science trying to understand how can you lower PRO-C3 levels. And I'm going to show you a little bit of those data. And of course, just high level, this is cancer-associated fibroblasts to the left, grown in culture. They are, if you stimulate them, they really produce a lot of type 3 collagen. If you block that with a TGF-beta blocker, then you can lower that. And to the right, you can see a serious red staining, and you can see really there are tons of collagen being produced, fibroblasts being active in this. Yeah, so of course, the PRO-C3 you saw before measured in serum was the reflection of active fibroblast. And then we can look at tons of compounds in human primary fibroblast and cancers to look at these levels. So, of course, this is one of the central things of our biomarkers. We want to link a cellular activity to a serological measurement. And so we start understanding this in preclinical settings, and then we understand it in clinical research, really to provide value in drug development. And so what we do with these Garnet jar cultures, we take fibroblasts, we activate them and become myofibroblasts. We used to just to activate with TGF-beta. Now we found a much more fancy cocktail, so we have different stimuli. So it's not just TGF-beta driven. We measure fibrogenesis. And then do analysis of that. We measure at many days to see what is the effect of given compounds on fibrogenesis. And as an example of this is against Garnet jar. It's hepatic stated cells, it's lung fibroblast, and then it's also intestinal fibroblast. And this is controlled, and then it's a fibrotic cocktail. And then you can see that the levels of PRO-C3 here as compared to controls is elevated many times. So it's a relatively robust system to assess fibroblast activities that our translational team is doing. Does it work? Well, nintedanib is of course was the first compound to be approved for IPF, idiopathic pulmonary fibrosis, and it really worked very nicely in this primary human lung fibroblast model. You can see there's a dose-dependent decrease with nintedanib on PRO-C3, and there was just a recent positive IPF study, phase 3 from United with tapostanil, and interestingly, there's a dose-dependent effect on on type 3 collagen formation, showing there's really an effect on the fibroblast in the lung. And Diana is going to talk much more about beautiful lung data in a second. So of course, in different settings, we measure PRO-C3. We do them in scarlet jar cultures. We measure them in liver models such as the carbon tetrachloride induced. We measure MASH patients. You have seen that. We do a range of different chronic kidney disease models, such as the different models in there. We also use them with colonic fibroblast, and of course we use in the BLIO model where we look at fibrosis. Okay, so with all these things, in particular the cancer cells, a cancer experiment showing if you have a high level above medium of PRO-C3, you have a between 3 and 7-fold increased chance of dying. Of course, if you have an important biomarker, possibly we can use them for much more than that. I'm going to show you some unpublished data that we think are reasonably fantastic. So I'm going to show you, I mean, can we help those patients actually? Could we find the reason why PRO-C3 is elevated? And so we have measured PRO-C3 in many samples, and then of course we wanted to see which genes, which pathways are responsible for this. Because normally when you do target discovery, you have a group of patients that are controlled and case, and that is the endpoint of the disease. And we wanted to see not the endpoint of the disease, but the process of getting there. Because PRO-C3 is not how many fibroblasts you have. No, it's how active are they. And so we wanted to see what is the pathways associated with disease activity. And so we have a wonderful cohort for this at Nordic. We've been doing research for more than 30 years, so we are following these actually 20,000 Danish women, and we have serum on these on more than 6,000, so we have full medical history because we have fantastic history for that in Denmark. And then we asked one question: what are the genes related to PRO-C3? And of course, we did a Manhattan plot, and there was only one gene, and it was big H3. So it's also called TGF-beta induced protein, but it's not TGF-beta. It's downstream of TGF-beta. And we were surprised. I had actually expected this to be a true Manhattan plot with many skyscrapers, but there was only one skyscraper. And then we took one of our models, and then we added big H3, and you can see when we add big H3, we actually have a dose-dependent increase in PRO-C3. And then Kim's group did a wonderful experiment to the right. To start with, the control cells, they add big H3, then there's 10 times more PRO-C3, then they add the antibody and block it, and then they lower that. That's not rocket science if you block the protein. But to the left, we actually hit with TGF-beta because big H3 is TGF-beta induced protein. So vehicle, and then TGF-beta induced, and then this antibody that is working downstream of TGF-beta is actually completely blocking fibrosis. We think this may actually be able to help patients. That's a different story. I think it's a wonderful way of linking pathways to unicorn biomarkers that we understand much more about that. So just a little bit of summary before Diana will take you on a roller coaster on lung and liver here. So around 40% of all deaths are linked to fibroblast activity. It's a key driver for disease progression. I think we need, and I'm glad we have the collaboration with Roche Diagnostics, because we need that to be happening in a normal clinical setting. You know your diabetes status, you know your cholesterol level. I think we should know our fibroblast activity level. This is just the start. If we develop biomarkers based on biological hypothesis, I think we maybe better make sure that they reflect the underlying activity of disease. Unfortunately, it takes a long path to develop a biomarker from an idea to an assay to a real assay to a technical fantastic assay to regulatory approved and then putting on a platform. It's called the biomarker valley of death, and I'm going to talk more about that. Then of course the translational link. I think it's very important that we can link the biomarkers back to cellular activities, because then we can find compounds, pathways that are modulating that. And I think that that provides us with an increased understanding of how it works. And then PRO-C3 is a direct reflection of fibroblast activity and can be used, of course, to diagnose and monitor patients over time. But Diana is going to show you much more about that. Diana?
Fantastic. So let me continue the talk and data from Morten, or presented by Morten. So I'll take you through some of the tip of the iceberg data that we have in the hepatic space for hepatic diseases leading to fibrosis, as well as the pulmonary space, where looking into populations who develop pulmonary fibrosis. So if we start out with the liver fibrosis part, then if you look at the slide here, or the data here, then you'll see, of course, we know that the extracellular matrix plays a big role in chronic liver diseases. And there's different stages of liver damage that can be occurring in patients. So ranging from normal liver, normal healthy liver, and then turning into a steatotic liver. Then some patients then progress to having liver fibrosis, and then at the end stage it becomes cirrhosis, and even some progress into hepatocellular carcinoma. And if you look on the bottom to the left, then you see here Mason trichrome staining, showing that from stage 1 to stage 3 you have increasing amount of fibrosis here in blue, and in this case also steatotic areas in the liver, showing that really it's super important to understand the extracellular matrix in this type of indication. I think that's very well established in the field, that we need to understand liver fibrosis in this case in different stages, ranging from this F0 to F4 stage of fibrosis. Next slide. And then you've heard a couple of times, this is something that we have done within the liver space, really changing the field, hopefully, for patients, that we have transferred the PRO-C3 onto the Lexis platform that you heard about. So it has a CE mark now since May, and also an IVD approval in Europe for significant and advanced liver fibrosis, as well as cirrhosis in MASLD patients, so patients with metabolic associated steatotic liver disease. And this is what we published in 2019, some years ago now. It can be used hopefully very soon with patients around the world. Well, in Europe first, and then hopefully around the world later on. Here you also see the ADAPT algorithm. It includes PRO-C3 as the main driver of the algorithm, together with age, diabetes, yes, no, and platelet counts. So we've established many collaborations to first of all develop this algorithm, and also verify this the use of this score in multiple cohorts of patients with chronic liver disease. Next slide, please. So again, I'll just show you tip of the iceberg data, and the first set of data I'll go through is how we are using PRO-C3 in the ADAPT score as a mark of staging. So really, as Morten mentioned, PRO-C3 was really designed to be a disease activity biomarker, but it also has a strong link to states of fibrosis in patients with chronic liver disease. This is the data we published in 2019 to develop the ADAPT score. So we had a discovery cohort and a validation cohort, and looked at the ADAPT compared to PRO-C3 as a single biomarker, FIB4, APRI, and NAFLD fibrosis score, which are some well-established scores within the hepatic field. And we actually saw, it's a bit small, I think, here for you to see, but the ADAPT score had an AROC of 0.86 compared to the other scores and single marker, which were lower for finding advanced patients for the discovery cohort, and in the validation cohort it was around the same. ADAPT had an AUC of 0.87. And again, this was done with wonderful collaborators from Australia and UK, and also in Japan and Denmark. Next slide, please. Then together with the Litmus Consortium, among many others, many scores that were established to do head-to-head comparison, ADAPT was, or PRO-C3 was measured, and then ADAPT was calculated. And then this is data published 2 years ago from the Litmus Consortium, and showing the head-to-head comparison here with this different scores. You see here the dotted line is the scores and single markers that reached above an AROC of 0.8 and actually were then outperforming FIB4. This was done by the SOMA signal and also ADAPT score. So I think in a big consortium like this, it's also been shown that the ADAPT score could perform very well for finding advanced patients in MASLD patients. Next slide. We also tested the ADAPT score in an Asian cohort, This is the data you see here. We've done few cohorts, but this is the one we published in 2022, showing that the DAP score had a very nice AUC, sorry, of 0.86 compared to others like PRO-C3, NAFLD fibrosis score, FIB4, BART, and ABRI that had lower AUCs for finding advanced patients or advanced liver fibrosis patients in this Asian cohort of more than 800 patients. So we know it can also be used in Asian population. We did this with several Chinese universities together, also with Westmead Hospital in Australia. The next slide. Then if we shift gear a bit, then we've used quite a lot of efforts on trying to understand how PRO-C3 can be linked to prognosis in patients. And in this case, patients with alcohol-related liver disease and experiencing decompensation. So this is a Danish cohort, single center cohort of almost 500 patients with full spectrum liver fibrosis F0 to F4, showing that actually when we measured PRO-C3 in these patients at baseline, you will see here on the right in the graph, the patients above the cutoff that we set for this population versus those in blue who are below the cutoff, so high versus low. We actually saw that there was a hazard ratio of 7 for patients experiencing decompensation in the high group compared to the low group. And actually what is also interesting to really see here is those patients who are in the low end of PRO-C3 at baseline, so having low disease activity, there were very few that actually experienced an event such as the decompensation. So it really matters to understand the activity of the disease. And actually we compared it to fibrosis stage, as you see here in the table, which we know is all linked to outcome and been shown by multiple groups around the world. Then actually we saw that this the PRO-C3 as a single biomarker actually outperformed in relation to hazard ratio, the fibrosis stage. So it was 7 versus 5. So it's really important to really monitor the disease activity in patients. Next slide, please. We also in the same cohort looked at different stages of fibrosis, Kleiner stage at baseline, and actually saw or looked at the prognostic capacity of PRO-C3 in those different stages. And if you look at the hazard ratios here, you see the hazard ratios are per change in PRO-C3 based on nanogram per mil measurement. And actually we saw that the hazard ratio dependent on the nanogram per mil change was the same across different fibrosis stages, indicating that really it really doesn't matter if you're an early stage patient or more later stage patient. Really the activity has a big role in the understanding the prognosis of patients with chronic liver disease, in this case ALD patients. Next slide. Then also actually very recently we have expanded this and to trying to understand not only fibrogenesis by PRO-C3, but also how does it look when we combine it with a fibrolysis marker such as CTX3. So it's really important to, of course, understand both fibrogenesis and fibrolysis because it's really the balance that will tell where the patients are heading. So actually we saw that both PRO-C3 and CTX3 were prognostic for liver decompensations in patients with alcohol-related liver disease. But when we combined the two, then we had a higher hazard ratio. If you look to the table, you'll see that those who had both high PRO-C3 and high CTX3, which is the lower bar here, we had a high hazard ratio of 18 for being prognostic for decompensation in ALD patients, which was lower in the opposite groups of having low, or the mix of those two. So really having high extracellular matrix remodeling really was highly prognostic for decompensation in ALD patients. This, I think, will be very important for the future. This is a PRO-C3 webinar and party, but really I think for the future we need to understand even more aspects of the disease, because these are complicated diseases that we really have to carefully identify and also look at the given mode of action of different intervention, and we need the biomarkers to understand the mode of action and which targets they are hitting. Next slide. And then, of course, PRO-C3, or not of course, but it's really something that we're very proud of. But PRO-C3 has been used in multiple clinical trial studies. I'm sure some of the people on the webinar here have seen previously some of these data, but really PRO-C3 has been used in phase 3 studies in patients with MASLD, or MASH, I should say. First of all, on the left, you see how PRO-C3 has been used in the Maestro-NASH trial from Madrigal, and THR beta agonist that has been tested here in this phase 3 study, which was a positive trial. Actually, it was shown here that PRO-C3 could be reduced by up to 23% compared to placebo patients by the resmetirom drug here. And on the left, on the right, you'll see semaglutide, which was just approved by the FDA. And I should just mention resmetirom was approved last year by the FDA. This year, semaglutide, the GLP-1, was approved in the Essence trial, also phase 3 study, showing here that PRO-C3 could be reduced up to 17% by semaglutide compared to placebo. So this data really shows that these drugs are modulating fibroblast activity and seems to be downregulating the activation of fibroblasts, which of course is important in these patients with liver fibrosis. Next slide, please. Then if we go to the phase 2 studies, PRO-C3 has also been measured in quite some of the big trials in this space of MASH. For example, with efruxifermin from Akero, survodutide from Boehringer Ingelheim, and tirzepatide from Lilly. Actually, they have also shown that they can reduce PRO-C3 and fibroblast activity with their different doses of their interventions compared to placebo. And some are shown here in nanogram per mil, some percent. You see, for example, survodutide, it's more than 20% reduction. For Lilly and Akero with tirzepatide and efruxifermin, it's nanogram per mil, but you see substantial significant reduction with all these different therapies that are having different mode of actions. So again, indicating they have antifibrotic effect or antifibrogenic effect. Next slide. That was the tip of the iceberg data on the hepatic diseases. But if we move then forward, I'll go a bit into the pulmonary fibrosis field. If you go to the next slide.
Diana, I'm going to allow a few questions because you have been bombarded. And there's some that's related to liver fibrosis here. And I'll just take a few. So one question is from Laurent. Can you comment on cold versus hot fibrosis across these different topics? But I think you can start in liver.
Yeah, this is a wonderful question, I think, and this is something that is on top of our minds at the moment. Because, as I mentioned, these patients they have a complicated disease. There's multiple. It's a multi-organ disease, especially in the case of MASLD. But I think for most of these, they involve many different organs and. We know from the oncology field that in tumor fibrosis that some patients develop this desert type of fibrosis in the tumor, and therefore it's difficult to really get in, target the tumor cells and really target that fibrosis. And we think that's the same in patients with chronic liver disease. You get this desert or cold fibrosis in the case of the liver, with potentially also in the lung and other organs. And that is tough to target. So we have developed some markers that we can use to understand these immunology-related processes of hot and cold fibrosis. So we think this would be important to use in the future for endotyping patients. Do they really have more hot or more cold fibrosis type? And how do you address these processes? Or if you have cold fibrosis, how do you then activate the fibrosis again? Because you need activity around the fibrosis part to really resolve it.
Can I add to that? So I think, so first of all, Diana and Andressa just did a wonderful paper called Hot and Cold Fibrosis in Journal of Hepatology. We'll be happy to send it, also freely downloadable. But first of all, I think we need to accept that not all type of inflammation is bad. I think we need some of it. And I think when you see CTX3, when you see fibrosis resolution, and that must be the action of some of the good inflammation. We need that good inflammation to have fibrosis resolution. Then there can be different types of bad inflammation. We can debate whether it's good to have M1 or M2 macrophages, for example, and there are specific biomarkers for that. I think it's really an important part. Then there are some tough questions, Diana. What is the circulating half-life of PRO-C3?
I don't think we've defined specifically because it's a tough one to do, but we know collagen fragments in general do not have a long half-life. And I think that's also part of so they filtered through the kidneys into urine. So you actually find a lot of collagen fragments in the urine and can be measured there also. But I think that's also part of the activity part, that this are fragments that are generated continuously in tissue where you have the pathology occurring or the injury occurring and activation of cells and production of extracellular matrix. And then this is reflection of what is happening. That being said, it's not like PRO-C3 just fluctuates like CRP. In the same way, we've done studies like this showing that it actually reflects something on over a longer time period. But it also is a benefit in relation to that actually it's a quite dynamic biomarker that you can use to find, if you want to look at effects on PRO-C3, you can find them fairly fast using this PRO-C3 test in your study.
Then there's a question on how does PRO-C3 correlate to the ELF test? I have a few comments on that. So first of all, we started with the half-life. I think it's important that some biomarkers actually are dynamic, such as a pharmacodynamic biomarker. And ELF was originally designed to be a diagnostic biomarker, and in fact, hyaluronic acid is being was put into the ELF algorithm. It's in the original papers because they wanted a stable algorithm that was not responding to therapy. And I think when we are debating reasonably likely surrogate endpoints and those, I think it's important that all other biomarkers that are reasonably likely surrogate endpoints are actually not composites. And I'm a little worried that resmetirom, that was the first approved, actually ELF is not responding to that. Of course, ELF is responding to GLP, so there are some things we need to understand. If we look at PRO-C3, how does PRO-C3 correlate to the entire ELF test? That's one question. I think the Diana has the right number for that. She'll answer in a second. But I think it's more interesting, what is the individual correlations of PRO-C3 to hyaluronic acid, to TIM1, and to P3 and P? And then even if we look at the correlation to P3 and P, that's above 0.6, depending on where you are in the curve, if you are actually a healthy individual compared to if you are a disease-affected individual, it's actually different processes of collagen that is involved in it. So it's actually different correlations on the curve. And so I'm sure Diana can remember what the exact correlation is with PRO-C3 to ELF. At least I gave you 30 seconds to think of it, Diana. Do you know that?
Well, I did think about it, and I think we're in the range 0.3, 0.5, something like that. There is a correlation. We are working on some of the same processes, but we're just measuring PRO-C3 as this S2 cleave fragment that is very dynamic. And at least from our investigations, we've seen that PRO-C3 is can be more dynamic in some of the studies we looked at for PD effects, pharmacodynamic effects, because it has this design that it has to be only that fragment released by this process of ADAMTS2 cleaving of the P3 and P molecule. And again, I think you can correlate to each each single biomarker in the ELF score, and you get a different number. But we do see on the clinical trials, they measure ELF and PRO-C3, we do see that they do have correspondence to each other, which I think is just a benefit, that we can look at this in different ways and see, like AST, ALT, we're modulating these as well, that we have multiple non-invasive tests that are pointing in the same direction. But yeah.
Anna, I just allowed a little bit, and that was already 5 minutes. Lung fibrosis, it is.
Yes, thank you. So I'll talk about pulmonary fibrosis again, tip of the iceberg data. We've tons of data in this space, and also we're moving into interstitial lung diseases overall and other indications, pulmonary arterial hypertension, and also CTD, ILD. So we're doing many different studies in pulmonary fibrosis. But I'll talk much about idiopathic pulmonary fibrosis, IPF, if you go to the next slide, because here we establish the PRO-C3 biomarker quite substantially.
Yep.
I'll start out like in the liver. Of course, fibrosis is very relevant in the case of patients having pulmonary fibrosis, because we know a lot of this is really a big problem in IPF patients, in interstitial lung disease, and overall there's a large accumulation of the fibrotic tissue in the lung, and then at some point then the lung decompensates and then the organ fails. So we have a lot of extracellular matrix being produced, fragments are released to blood circulation, we can pick up the fragments in a blood sample. Again, also PRO-C3, but also many other markers that we're investigating in this. And actually for this space, the primary endpoint for IPF is pulmonary function test. I really think there should be a large focus also on the fibrosis part, having fibrosis biomarkers. And that, of course, I know the field is working hard on, but really to modulate the disease, I think you need to have fibrosis-related biomarkers. It cannot only be pulmonary function tests, because this is a result. The pulmonary function test is really a result of many different factors playing a role in patients with pulmonary fibrosis. So if you go to the next slide. I think this has been mentioned a couple of times. It's important to understand formation and degradation, and PRO-C3 is measuring formation of type 3 collagen, but we also measure other collagens for formation degradation, and also something for the basal membrane, from in situ matrix, looking at epithelial damage and so forth. But if you look at the graph here on the right, in the very top, you'll see, and it's a bit small again, sorry for that. If you look at this graph, you'll see the blue patients of IPF. These are the fast progressors versus the group in gray, which is the more slow progressing patients of IPF. And then on the dotted line, you see the healthy reference range. Then you see clearly PRO-C3 and active fibroblast activity, or active fibroblasts are highly elevated in progressive IPF and IPF in overall. But we also see that progressors are higher in PRO-C3 compared to the more slow progressors of IPF. So we can actually separate the disease activity of patients who progress faster and show that PRO-C3 is prognostic for change in lung function and also in mortality. So this is based on FVC, forced vital capacity, and mortality, this as a composite endpoint and definition of the patients. And we also see for other markers like the PRO-C6, which is also fibroblast activity, marker of endotrophin, higher in progressive IPF compared to less progressive IPF, and highly elevated compared to healthy controls. And then other markers I'll not go into today. If we go to the next slide. Then also in another cohort, so I forgot to say the previous data was in the profile cohort from the UK that we did some years ago, together with GSK and Royal Brompton and Nottingham. This is the data you see here is actually from a pure Danish cohort from the main region. It's Herlev and Gentofte Hospital and also Aarhus Hospital. We then looked into these IPF patients and then looked at how high extracellular matrix turnover was associated to mortality. And if you look to the left, you'll see if patients at baseline had one biomarker that was high, then they had a certain survival probability. But if they had multiple biomarkers that were high, here in blue, you see that those patients had a lower survival rate. So it means that having multiple biomarkers with high indicating high ECM turnover, that was associated with an even increased mortality rate. And then on the right, this is the same thing that you see, or similar thing. This is number of high biomarkers at baseline and then linking it to forced vital capacity, seeing that those patients with higher number of high biomarkers at baseline, they actually also have a lower forced vital capacity at baseline, together also with diffusion capacity, DLCO. In the same way you see here, the higher the number of high biomarkers at baseline or higher ECM remodeling at baseline, then the lower the DLCO, the diffusion capacity. So we can see that these markers are actually linked to something that is very relevant in these patients, like lung function and diffusion capacity, which is also under lung function. But that's very important, I think, to have this link. If you go to the next slide. Then if we look at pharmacodynamic effects and PRO-C3, then we've done quite a few analyses together with different collaborators. Here it's with the JNK inhibitor from BMS, the CC19001, and they have shown that they have some effects on forced vital capacity with different doses of their intervention compared to placebo on the left. And on the right, you'll see PRO-C3, also PRO-C6, actually was modulated by the JNK inhibitor, especially one of the doses compared to placebo. And actually that corresponded to what you see then on the left bottom graph, that actually PRO-C3 increase or change from baseline actually corresponded to a decrease in forced vital capacity, whereas those who decreased in PRO-C3 and responded to the therapy potentially, those actually had increase in forced vital capacity. So it seems like it's a benefit to decrease PRO-C3, then you gain some of the lung function that was lost. If you go to the next slide. So this is another study in a phase 2 study of IPF patients. This is the Bioscience alpha V beta 6 beta 1 inhibitor from Pliant. Here they also measured PRO-C3 and saw that actually with the different doses of Bioscience that they had reductions of PRO-C3 already at week 4 and also week 12. And actually that also corresponded again to the proportion of FVC decliners. Or I would say it was linked. On the left, you see the proportion of FVC decliners. You see that with the different doses, they have increase in FVC over time. And that links to also having a reduction in PRO-C3. So this data also shows that decreasing PRO-C3 fibroblast activity has a link to increasing forced vital capacity, at least in phase 2. The next slide. That leads me to the summary. That was a lot of data, and again, we have much more data to do. Let me know if you want to just see even more data, if that's of interest. But what really we looked at was that fibrosis is, of course, evident in patients with pulmonary fibrosis and we really need to understand fibroblast activity and fibroblasts and hepatic stellar cell activation in these patients. And we also saw that understanding fibroblast activity is important in overall to evaluate and monitor patients that are living with fibrotic indications. I also showed you that PRO-C3 adapt can be linked to liver fibrosis state. We also know PRO-C3 is a single biomarker can be linked to fibrosis state, but it's improved with adapt in patients with chronic liver disease. I have also shown that PRO-C3 was related to outcome both in patients with hepatic disease and pulmonary fibrosis. So in the case of mortality and change in FVC and decompensation. And lastly, PRO-C3 can actually be modulated by intervention in both of these indications of pulmonary fibrosis and chronic liver disease, and hope to aid in decision making in early stages of drug development. And that was it for the hepatic and pulmonary fibrosis presentation of PRO-C3.
Thank you, Diana. One of my favorite papers is from Wuntan Jiao and the Nimble group. They looked at 1,800 patients with different stages of liver fibrosis. And unless you have late stage liver fibrosis, then you actually have more cardiovascular outcomes and you have more kidney related outcomes. So of course, patients should be treated as a whole patient, not just an organ. And that's why, of course, looking at cardiovascular disease these days is very important. I'm just going to show 3 slides. So PRO-C3, or I should say endotrophin, that we're measuring now, a sandwich assay on endotrophin. It's actually a real hormone. Many things are really pointing in the direction that endotrophin is a true hormone driving fibrogenesis. So I am interested in this because I think many have been looking at the link between wound healing and fibrosis for a long time. And the reason for showing this today is that we actually just agreed with Roche Diagnostics that we would be putting endotrophin on the Roche Cobas platform in the sandwich assay. Linking fibrosis and wound healing is important. So actually, type 6 collagen alpha 3 chain contains 12 von Willebrand A-like domains. It means that activated fibroblast, because this is produced by activated fibroblast, can activate wound healing without von Willebrand. It means that activated fibroblasts directly bind platelets, and platelet is the biggest source of TGF-beta and PDGF. And so that means it further aggravates fibrosis. So endotrophin is a measurement of super activated fibroblasts. And I just have 3 slides. One, we are celebrating another collaboration with Roche, which is going to move the fibrosis field forward, I think. And then secondly, if you are about to become afraid of fibroblasts looking at the PRO-C3 data, I think you will become really afraid now. So we looked at many studies, fantastic collaboration with BMS and others. This is my first and only New England Journal of Medicine publication, so of course we need to put that in. We measured PRO-C3 in 6 different studies, and it all showed the same thing. If you take the ones in the higher tertile, this is just tertiles, 70% actually have a chance of an outcome. That means we can identify 70% of patients that's going to have a fibrosis-related death in this disease. Later, Federica and the team took this into measurement of, I think it's 20,000 patients in that cohort, in kidney disease, and cancer, and diabetes, respiratory, different others, and the overall odds ratio was 2, meaning the healthy reference range of PRO-C3 is around 8. Many, in particular in cancers, at very, very high level. So the chance of dying if you have a high level is very high. There was a question in the chat: does it make sense to measure both PRO-C3 and PRO-C6? Of course, we would think so. It's really fibroblast activity. They are correlated, but it's not more than 0.5. So it's really important that it's 2 different types of fibroblast activities, and if they are high, unfortunately, there's going to be an outcome for those patients. Then Alexander and Federica did a lot of additional analysis, and this is in a Spanish cohort. So we measured really thousands and thousands of samples in PRO-C3. And it's interesting that if we just look at tertiles here, then in the upper tertile, above 15, the hazard ratio for dying is 10. It means if you have a high level of PRO-C3, I should just say if you have a high level of super activated fibroblasts that aggravate wound healing, then your chances of dying is 10 times higher for those that do not have. We have done many samples on this, and just 1 nanogram is actually giving us a roughly 10% increased chance of death. So it's really important with these. There's not enough time for that today, and it will be in the next PRO-C3 webinar. Weight loss alone does not change this. Pharmacodynamic modulation with weight loss drugs actually is doing it. So there's some very interesting things in the dynamics of obesity management. So I'm going to show you a few additional diseases, and then we have a discussion in the end. Just going to show you a little bit in kidney. There's a lot of matrix in kidney. That's a topic for another day, and Federica have developed wonderful markers for that. I just want to say that PRO-C3 is actually not particularly the best ones for kidney. However, we go to urine. It's my favorite. So serum PRO-C3 is not particularly good for kidney. If you look at urinary levels, then according to CKD stage, you can see that formation of type 3 collagen in urine goes up and the degradation goes down. So the balance is shifting as a stage of disease. If we look at the correlation of type 3 collagen degradation to outcome in stage renal disease, then there's a very big difference between low and high. This is medium cut, and it's correlated to tubular fibrosis. I think this is interesting. So Diana showed you briefly CTX3, our new fibrosis resolution biomarker that we have been working on with many collaborators. And this is the Canvas study with the canagliflozin compound and SGLT2 inhibitor. And what you see here is a dose-dependent inhibition of type 3 collagen formation and a dose-dependent increase in type 3 collagen degradation. Possibly that's why it's actually working on kidney function. There's a question in the chat: have we seen compounds where that's actually working? This is one of them. There are other examples. And also in bariatric surgery, really CTX3 levels go sky high. So if we look at kidney, by far the best biomarker, it actually outperforms GFR, and predicting GFR is looking at PRO-C3 and mortality. So if you have a disease and you have fibroblasts that's activating wound healing, this can be in the risk of cardiovascular disease, it can be in CKD progressing, it can be in all-cause death. The central take-home message, and thank you to all these collaborators, there's more than 20,000 patients on this slide. There's a sky-high hazard ratio rating from roughly 2 to 3 in outcome. So if you have super activated fibroblasts, that's a bad thing. I think we need really to put them on platforms and make sure that we cater to patients. It's fair enough, you know what your inflammation level is and what your cholesterol level is, but you need to know what is your level of fibroblast activity. Then a little bit of inflammatory bowel diseases. Joachim and team has done wonderful work, also together with Takeda in finding fibrostenosis, and and others. And there's really not enough time for that, but I just want to show you one study done with BMS. It's the ozanimod study, because it really shows, I'm going to be provocative and say that patients can actually feel the extracellular matrix remodeling. So this is a phase 3 study, and what Jürgen was doing was looking at the ratio between PRO-C3 and C3m, and both dosages of these are having a significant decrease. Another biomarker is neutrophil activity. Story for another day, FDA supported, but neutrophil activity, not number, was decreased. But this is really what's important, because this was responders to treatment based on a how patient function, feel, and survive scheme. And the ones that were remitting had highly significant decreases, but those that were not remitting were not. I know it's a little bit possibly overstating this, but it is, to me, suggesting that patients can be feeling the extracellular matrix. We've seen that in RA as well. We'll see it in a second. And it's important, at least it's important for response. And then, of course, that neutrophil activity is super important for IBD. There's been a lot of discussion in RA of a fibroid phenotype, and Anna and her team had really done wonderful data in showing that those patients that actually have a high level of fibroblast activity, quantified as PRO-C3, actually have less response. So if we look at the study, it's a study from Genentech. We've been looking at tocilizumab, a wonderful compound. And in this one, if we look at what is the odds ratio for responding if you have a low level versus a high, just medium cut, then the patients that have in both studies, in both ambition and radiate, for the ones that have a low level, the odds ratio of being a responder measured on ACR 50 is actually twofold. So fibroblast activity, again in RA, is super bad. If you have a high level, then you are a much worse responder. If we look at PRO-C3 in response to different treatments in RA, I think it's important that, of course, methotrexate, those that are taking that and those dosages is actually suggested that should be monitored, and you can see actually that on average there's a 17% reduction or increase in PRO-C3 because methotrexate is giving liver issues. And then others are adalimumab and toci is not affecting it, and tofat is actually having an antifibrogenic effect that correlates with preclinical studies. Last thing is systemic sclerosis. Really sky high levels in those that have early disease as compared to late disease. And seen in her team in skin, and there's just the tip of the iceberg, and it's actually Atlantic Rheumatology publication together with Oliver Ditzler. If we look at the area under the curve, just looking at the balance between formation and degradation, again, those patients that had a high level in that, with when we looked at stable versus progressors, the area under the curve is 0.85. So really a balance that is meaning something. I'm going to end up here, but just before we do that, we've started using PRO-C3 kit in a box, so we sell that. I think it's important we do that because I think if we need to enable other fibrosis researchers to assess fibroblast activity. PRO-C3 is the first one. It's for human use. That means it needs to be in human cells or in human serum samples. And there's a rodent assay coming in a second. And the next assay coming out is PRO-C6, enabling us to really link wound healing and fibrosis. I think we need compounds that are working on PRO-C6. And then, of course, as I started, having this much research is a super team effort. We saw a little bit of the data from the neuroscience group. Federica has been doing all the cardiovascular data. Yanni and Diana have been doing the lung and liver work. Jürgen is doing all the IBD work, Signe the skin work, Nicholas all the cancer work. And then nothing of this is happening without a fantastic lab that's measuring it. I just had one slide of the lab and a little bit where you saw the fantastic equipment that we have in there, and that's headed by Tina and our right wing Kishwa in that. I've not thought about production and other things, but it's a big team effort. And so, in summary, fibroblasts are central players in fibrotic diseases, and active fibroblast activity is linked to 40% of death in the Western world. Let's let's make sure we do something to quantify that fibrosis activity of patients, in addition to cholesterol and inflammation. PRO-C3 is reflecting fibroblast activity in real time, really providing a translational link, what happens in the tissue, and that we can measure in serum. Many studies have shown that it's related to outcome. I think it is super important that we measure something that's related to outcome. And then, of course, we can track fibroblast activity over a long period. That allows us really to think, should we be developing this compound or not? And then the last part is this. Today is a celebration. We've been fortunate over a long period with many fantastic friends and collaborators, and to take PRO-C3 from a hypothesis-driven approach, saying this is a particular fragment of the propeptide of type 3 collagen that we need to develop an assay for. We developed 30 different assays for type 3 collagen, and this was just by far the best one. And that allows us a good measurement. I see that we have a few minutes left, and there are tons of questions in the chat, and I'll try to ask a few of them here. I think the first one here is for you, Diana. Can you remind us of the role of type 3 collagen in the pathogenesis of interstitial lung disease?
Well, we know there's a lot produced during a progression of pulmonary fibrosis, and then really we think that there is type 3 collagen present in the case of healthy lung, but really type 3 collagen is one of the collagen that really expands or increases in amount, so to say, during fibrosis progression in patients with IPF. Again, I don't, it's not super well established with exactly the amount. I know there's a lot of RNA-seq data being done, and also we know type 6 collagen plays a huge role also in the case of pulmonary fibrosis. So really, I think type 3 and type 6 collagen are extremely interesting to understand, together, of course, with some of the proteins that are present, like elastin as well, that is present in the healthy lung. But we know that this is one of the collagens where the fold increase is high in case of pulmonary fibrosis.
Thank you, Diana. I'll take another question. Also, type 4 collagen has been implicated in glomerular disorders in kidney. Have you looked at type 4 collagen turnover in CKD patients? That's a good question. We've developed a range of assays. Tumstatin, for example, is the signaling fragment of type 4 collagen, sky high in different CKD patients. We've also looked at Alports and what is the collagen turnover in Alports, and there's some very interesting findings that belong to Frederica. But basically, I like to answer the type 4 collagen question because type 4 collagen is actually a good biomarker. Or it's not a good biomarker. It's a good protein, and we need to have type 4 collagen in the different tissues to allow the epithelial cells to carry out their function. Wow, have you ever looked at PRO-C3 in hematological malignancies? Of course, we've looked at PRO-C3 in multiple myeloma. There's a lot of bone fibrosis in multiple myeloma, and that's highly elevated in that. This one is for you, Diana. Is the protein fragment carrying the PRO-C3 epitope unique in human circulation? You're muted.
Sorry, can you repeat the protein signature?
Is the protein epitope unique in human circulation?
I think so. Yes, I think so. When we have selected targets for generating antibodies or raising monoclonal antibodies, we always check the sequence and the cross-reactivity potentially to other proteins. And there we've done the same for PRO-C3 and also all other markers. We always cross-check and select these specific epitopes that are specific for the given protein that we want to measure. So I think yes.
I think it's important. What are the differences between different platforms? Of course, we're using monoclonal antibodies, and of course we screen for the fact that the small 7 to 10 amino acid sequence that we use. But because it's neoepitopes, we also make sure that all our antibodies do not bind to the elongated sequence, and that makes them super specific. It takes a long time to develop, but that also makes them super specific. Then I just want to dig down into the question, because it's important doing collagen research, because there are 28 collagens, 46 different cytins, and they look very much like each other. So when we're doing this, it needs to be monoclonal antibodies. It cannot be polyclonal antibodies that have cross-reactivity to all the other collagens. It's really, we are getting bombarded with questions. I think that's very interesting. So I'm going to give you this last question, Diana, because this is a tough one. Are you ready? Do we have any insights on why distinct types of collagens are key in driving fibrosis in distinct specific organs? There's a follow-up question to that already. I think it's the meaning of this question is there different collagens that are driving different fibrotic diseases? What are the common denominators and what are the specific ones?
Yeah, I think that's a very interesting question. And we do think that some of these fragments can also be part of driving disease. And there's maybe some common sequences in some of these that are signaling to cells when you have, an abnormal level of these fragments. And another thing I could also mention, if I understood the question correctly, but also we target sometimes some epitopes that are specific for different organs. So we have like fragments that are specific for the lung, for the kidney, and so forth. But I do think that they can play a role in relation to disease progression.
Thank you. So that's a big question, right? Are biomarkers, are they just the consequence of disease, or are they the cause of disease? And I think if I'm going to be looking into my crystal ball, I think neutrophin may both be the consequence of, but also driving disease. But you'll see much more about that. We spent 1.5 hours together. Thank you for being part of our celebration where we have launched PRO-C3. Many questions have been asked. Make sure that I or my team members will be answering all those questions. I think it's important that we measure both fibrogenesis and fibrolysis. That's going to allow us to find the best compounds. And thank you for joining.